On exam number 2, I'm going to start with something like this, and you will have to give me that. Everybody cool?
Mm-hmm.
Right? Yeah. What? Oh, we're going to get to that too, and I'm going to show you 2 methods. There's the method that nature uses, and then there's the Ray Burke method. Which one do you think is better?
The one nature uses.
Show you my method. Okay, so now you all understand exactly what you're going to be responsible for on the exam, besides knowing all of these terms and how to use them. I'm going to give you this, and you're going to have to fill that in for me. Okay? And then the next thing we're going to have to do, and by the way, that's just completing a DNA molecule, because you all know that DNA is a double-stranded, anti-parallel DNA molecule with 4 nucleotides, right? Or 4 different types. Next, I went ahead and took this DNA strand and I copied it again. Only I made it a little bit easier for you because I split them up in groups of 3s. You see that? It's the same letters, same nucleotides, but now they're split up in groups of 3. Does everybody see that? Okay. And the next thing I wanted to do for you is tell you that each group of 3 is called I know somebody knows the answer to this.
Codon?
Yes, a codon. Each group of 3 is called a codon. Each group of 3 happens to be a word in this blueprint. Next, if DNA is inside a nucleus, remember it's a double-stranded molecule, you can't make the products inside the nucleus because the nucleus has tiny little pores in it. If you manufacture the product inside the cell's nucleus, it would be like building an airplane in your garage. And then now you can't get it out of the garage in order to go fly it. Or building a boat in your living room, right? So you shouldn't be manufacturing the products in the nucleus. Instead, you should take a copy of the plans and stick them through one of those little holes so that the product can be built out here in the cell. Mm-hmm. Does that make sense? Mm-hmm. So this little messenger RNA, mRNA, sometimes you'll see it as mRNA. This messenger RNA, we have to do something called transcription. You're going to have to be able to go from a DNA molecule to an mRNA molecule.